All right, so hi everyone. Thank you so much for joining our Flow Cytometry Data Analysis workshop. I'm really excited to have Michelle Belmont here today, who is one of our scientists at Proteintech who will be running the workshop for us. Before I pass it on to Michelle, though, I just wanted to share a few company slides to introduce Proteintech. So, Proteintech is a leading manufacturer of antibodies and immunoassays. We have a very expansive portfolio of antibodies. We also offer antibody labeling kits, ELISA kits, IHC kits, and cell separation kits. Within our Proteintech brand, we also have our humankind branded cytokines and growth factors. And all our proteins are expressed in human HEC293 cells, so you can be confident that they have native folding, post-translational modifications, and glycosylation. We also have our Chromatech branded nanobody-based reagents, which are based off of alpaca nanobodies. And due to their small size, they are ideal for immunoprecipitation, immunofluorescence, and protein purification. I also just want to take a moment to celebrate the success of our customers because our customers' success is also our success. And we're really proud that we recently reached over 200,000 citations of our Proteintech products worldwide. And we're also really proud that we were the winners of the Researcher's Choice Award from CiteAb last year, showing that our customers really do value our products. So with that, I would like to introduce our speaker today who will be running our workshop, Michelle Belmont. So Michelle received her bachelor's in science in molecular biology from Loyola University here in Chicago. She then went on to do her master's at Loyola in infectious disease and immunology, where her research focused on natural killer cells in pregnancy. Michelle has a lot of flow experience in both academic and industrial settings, and she's currently a scientist here at Proteintech in our flow cytometry team. So during her talk, if you do have any questions for Michelle, please input them into the chat or the Q&A box, and we'll address them at the end. So with that, I want to go ahead and pass it off to Michelle. Thank you so much for being here and for sharing your wisdom with us.
Yeah, I'm really excited to share some more about my knowledge of flow cytometry with everyone. So I'm going to go ahead and share my slides with you. Get my laser pointer out. So I'm going to share today some tips and tricks for getting the most out of your flow cytometry data and a little bit of setting up your experiment to set yourself up for success in analysis. So since you're here, I'm assuming you have some sort of use for flow cytometry in your work, but just in case, I wanted to highlight some benefits of this assay. So you can use various sample types for flow cytometry, bodily fluids, any sort of tissue suspension, blood, bone marrow, or even a cell line. And you really don't need very much of what you're sampling. Oops, okay. Yeah, you really don't need very much of what you're sampling because flow cytometry allows you to detect infrequent and rare cell populations within your larger sample. And you can pare down your cells into multiple subgroups without separating them before you run. So that is pretty powerful. And also compared to other protein identification methods, it has high throughput, it's fast, and it's quantifiable. So you can find out a lot about your cells of interest with just one experiment. And since every experiment is different, I'm not going to go in depth on protocols here, but this is a general workflow that you might see where you have extracellular staining. Sorry, my light just went off. Where you have extracellular staining and you could run the cells directly after that. Or if you have intracellular targets that you're also interested in, you could fix and permeabilize the cells and stain either the cytoplasm or the nucleus for other targets. And then once you have detection, you are ready for analysis. And again, the goal for analysis is going to be a little bit different for everyone, but there are a few main reasons that you will want good analysis results. One is to understand more about your cells of interest. Like I said, it can give you a lot of information about these cells with just one experiment. You can also use the analysis to validate results seen in other applications like Western blots and ELISAs to make sure that your protein is really there. and also to produce a clear story, especially if you're close to publishing or if flow cytometry was requested by reviewers. And again, you need to confirm results seen in other applications. So in this talk, I want to walk you through some helpful tools and experimental setups that will help you save time in your analysis and avoid having to redo experiments. So if you set yourself up well in the beginning, you're going to have an easier time in your analysis. And in order to really set yourself up for success, it's good to know your flow, specifically your flow cytometer. And so flow cytometers can measure both fluorescence in the visible light spectrum and via the bright field. So even cells not stained with anything fluorescent can give you data about your cells. And so in the machine, cells will flow past lasers, and when the light hits each cell, some light will be able to go through the cell and hit a detector, and that will give you forward scatter, or a measure of the surface or size of the cells that are going through the machine. And some cells have more complexity within them, which causes the light to bounce off of granules inside the cell or large structures, and it will bounce as it's showing here, and that is a measure of side scatter, which is scattered at a 90-degree angle to the laser beam, and that will tell you about the complexity of the cells that are running through the machine. And so, for example, if we run human PBMCs through the machine, we'll see something like this, where the forward scatter, or the size of the cells, is on the x-axis, and the side scatter, or the complexity, is on the y-axis. And we know that monocytes are larger and more complex than lymphocytes, so we expect them to be higher in both of those parameters, and this is without any fluorescence. But when we look at a cell population that's more homogeneous, such as a cell line, there's really only one main cell population. So in both cases, forward and side scatter allows us to get the cells we want without junk and debris. Since dead and dying cells tend to be smaller and less complex, they might fall down towards the left corner of this axis, and then you can gate them out from the very beginning without using any channels to take out any debris. And so we can take what we want and leave the rest without using any fluorescence channels. We can also get rid of doublets, or cells that are stuck together, using these light scatter parameters. So in this case, let's say we take the lymphocytes in this graph on the left, and as each of these cells moved through the laser, they produced a curve known as a voltage pulse. So as the cell moves through, the pulse will start. When it's at the center of the laser, it will reach a height, and then as the cell leaves the path of the laser, it will come back down. And so we expect that a single event, or a single cell, would have a relatively proportional height to area ratio, and so those cells would fall in a diagonal line. However, if you had two cells stuck together, say, the peak does not go back down to zero before the laser hits the second cell, leading to a second peak, and that area would be then larger than the height, and then these cells would fall up here. So we can gate out any events that are two cells stuck together, leaving cleaner data and something that you can be confident in. And your machine will automatically collect this data on forward and side scatter while it's collecting fluorescence data. And so each cytometer has a different configuration. The general idea is still the same. So the cells will flow single file through the lasers, each at a specific wavelength. So this image is just depicting one laser, while most flow cytometers will have more than one spread out over this single cell sheath. And so fluorophores that label the cells that you use in your staining are excited by the lasers and will emit photons at a specific wavelength. And we already talked about forward and side scatter. So forward scatter tells about the size, side scatter is the complexity. So there's detectors for those. But also these photons will pass through a set of dichroic filters, and that will function as mirrors that lead to specific detectors. And these vary based on the configuration of your machine as well. So it's important to know what your machine is equipped to look at. But each laser can have different bandpass filters, which reflect light within a narrow range to these detectors. While photons with wavelengths higher than this specific range will pass through and go to more filters until it reaches the filter that reflects it. So in this simplified case, the photon in this pink line, the wavelength is too long to be reflected by these two dichroic filters, but it falls within the narrow range of this filter, and it will go into this fluorescence detector. And as I've mentioned multiple times now, each cytometer has a different makeup. So for example, this filter will reflect light to a detector that's between 515 and 545 nanometers, with the peak collection at 530 nanometers. While any photons that are higher than 545 will get transmitted through this filter to filters with higher nanometer wavelengths. And this is why it's important to know the emission spectra of the fluorochromes that you're going to be staining your cells with. And that will help you make sure that you have a filter and a detector for that specific fluorochrome's emission. And so each detector on your machine will have a filter best suited for it. And that's very important when it comes to building your flow panels. So similarly, each fluorochrome can absorb and emit light energy over a specific range of wavelengths. So in this case, I'm showing you our dye Corelight Plus 647. It absorbs light through this range of wavelengths, with the peak absorbance at 647 nanometers. And then the photons will be excited and emit light at a wavelength at a peak of 667 nanometers, but it can emit at anything along this range. And so if you start off with a panel design that works well together, it will be easier to tell your story once you've run your experiment. So for example, say you have a target in this Corelight Plus 647, but you have two other options for a second target. One is in Corelight Plus 405, and one is in Corelight 594. You'll want to select the antibody for your second target that has less spectral overlap, or even no spectral overlap with that first color. So we can see the 405 is completely separated from the 647, whereas the 594 has emission overlap with the 647. So it's more difficult for the machine to figure out which channel or which detector that fluorescence is coming from. And so, like I said, this is why it's important when you build your panel to check this overlap of your desired fluorochromes and to avoid using the ones with high amounts of overlap. And there are ways to get around this. And that's where your controls come in. So say you have to use all three of these colors in one panel, there are controls that you can use so that that allows you to do that. And so when you run an experiment on flow cytometry, your controls are just as important as your actual samples, and they can make a huge difference when you go into analysis. So the most important control in this case is compensation, and compensation will help determine the spectral overlap from each of the fluorochromes in your panel, and which signal is due to which fluorochromes. So in that example I just showed you with the spectral overlap, compensation controls will allow you to use all three dyes in the same experiment. And in order to do this, you should have a single sample for each of the dyes that you use in your panel, and each one should be stained with only one color. And the antibody used for these single stained compensation controls should always be the same antibody that you use in your panel, so that the dye is correct and the machine can make sure that a positive signal from that dye is correct. And so each machine is different, but compensation controls might also let you adjust the voltages before you run your samples. So that way your cells will be as bright as possible in the channels that you're looking at. But you would have to do more research on your specific machine on how to do this, because like I keep saying, each machine and each experiment is different. And you can use beads or cells for your compensation controls. I recommend beads personally because they're designed in such a way that half the beads will pick up your dye and half of the beads will remain negative. Whereas if you use cells, you might have some targets that are highly expressed, so you won't get a negative population in that channel as a control. And some cells may not have enough of a target to show what a true positive in that channel would look like. So for example, this is compensation using beads, where half of my beads picked up this PE dye that I was using and half remained negative. But when we look at the ECD channel, we can see that this PE stained beads are also showing a positive result in the channel directly next to it because of the spectral overlap that I was describing. And so the goal with compensation is to tell the machine that these single stained beads are only positive in this channel. And so we want a calculation that will bring this positive population to be single positive for PE and align with the negative beads. And so once that calculation is complete, we can see that the machine can tell which fluorescence now is coming from the PE channel. And like I mentioned, the machine should do this calculation for you. You can also do it in your analysis workspace. So in this case, I'm using FlowJo, but almost every analysis software should have a compensation tool. So once I dragged my compensation single stained controls into the compensation group, I can use this compensation tool. And that way I can look at all of my beads and make sure that the positively stained beads are highlighted so that the analysis software knows what a positive result in that channel looks like. And so once you highlight all of your positive beads for each channel, you can apply that compensation to all or a subset of your samples that you need that compensation for. And this will create a compensation matrix where all of the calculations are done by the computer. But sometimes computers are not perfect, so there is a way to adjust this, and that's by editing the compensation matrix. And so here I have an example of compensation that was calculated, but was not calculated properly. And so we can see that there seems to be two positive populations in this quadrant 2. I know personally that not all CD3 positive cells are also CD4 positive. There should be a larger population in quadrant 1. So when I go back to my single stained compensation control samples and use the method I showed you with aligning the negative and positive cells for that channel and making sure there's no overlap into the channel that it is bleeding into, we can have a graph that looks better. And this quadrant looks more accurate to reflect the cells that should be there. And I want to stress again how important it is to have your single stained compensation samples and only use those to adjust your compensation matrix to avoid any bias when looking at your stained samples. So you definitely don't want to look at these when you're adjusting your compensation, because that could lead to populations that are not actually there to look like they are. And as I mentioned previously, it's really important to use the dyes that have emissions that are further from one another on the visible light spectrum, especially if those two dyes are going to be staining the same cells. And that way you can separate out true double positive cells from cells that only express one of those two markers. Another way to avoid bias when you're gating is to always gate first on an unstained sample of some kind. So unstained samples will reveal any autofluorescence that your cells might give off. And it also allows for you to set your gates where there's no staining so that you know that any positive signal you see from your stained sample is correct. So in this example, the gate for the Coralite plus 750 channel was drawn on this unstained sample, so that we know that when we apply this gate to our stained sample, we can be positive that all of the cells in this gate are truly positive in that channel. Sometimes dyes, though, will have some background, and that's why isotype controls might be more accurate than an unstained control, because it accounts for the dye molecules that may be unbound to your antibody or left in the sample after your wash steps. So knowing the isotype of your antibody and having it conjugated isotype control is an easy way to account for the background fluorescence from the dye that you're using. And so the same method can be used, and this is the same sample, but instead of gating on an unstained sample, I gated on a sample that was stained with a conjugated isotype control so that we can gate out any cells that might be positive for only the dye and not for the marker of interest. And so I'll show you when you apply your unstained sample gating to the isotype control, it adds about 1 to 2% of positivity. So if we use the unstained sample to gate on our stained sample, we have about 20% CD19 positive cells. But when we use the isotype control, it gets rid of about 2% of those that were false positive simply because of the dye latching onto the cells non-specifically. But how would we go about this in larger panels where we might want to be a little bit more precise with drawing our gates? That is where we can use FMO or fluorescence -1 controls. And these controls are stained but all but one, so fluorescence -1 of your samples. And so in this case, I'm looking for STRO1 positive stem cells. And so I stained a sample with every marker in my panel besides the anti-STRO1 to find that rare population. And this rare population happens to be CD45 dim and CD34 positive cells. And this is a rare population in the blood. So it's important to use an FMO control in cases like this. So I did the gating on the FMO, finding that rare population of CD45 dim, CD34 positive cells. And then from that gate, I can draw my gate for STRO1 so that when I do apply this gating to my samples stained with anti-STRO1, I can be confident that all of the cells that fall within this gate are truly positive for that marker of interest. And there are two tools that can make organizing your samples within your workspace easier. And the first is groups. So groups allow you to organize your samples so that you can apply gates to all samples in that group. So in the previous example, I could group together my FMO and my STRO1 and apply the gating from the FMO to the STRO1 stained sample without applying that gating to every sample in my workspace. And you can create more than one group, and the samples that you have in your workspace can belong to more than one group so that one gating method doesn't get lost in all of the gates that you have in one sample. You can separate the different gating methods by group. And the second is using keywords. So keywords allow you to attach descriptions to your samples, and this acts as metadata and can help with organization and also with statistics. You can add a keyword and apply subgroups to it. So as an example, say I have a keyword that I want to organize my sample by which patient that sample belongs to. So I can create a keyword that says patient, and then I can label my samples patient 1, 2, 3, etc. So that all of my samples from patient 1 have that metadata attached to it so that I can trace it easily. So now that we've gone through organization of your data and applying gating from unstained or FMO controls, now it's time to get your data ready for publication or whatever message you would like to get across with your flow data. So first, I want to discuss the various graph types that you can create when you're analyzing. And one option is to display your events by density or where they fall within a certain parameter. So we can see here that the red area is where most of the cells fall within this population. Contour plot does the same thing where there will be a population that is clustered within this contour, but it doesn't show the actual events. And these two types of plots are very common in publications, but you could also use density plots and zebra plots to reflect this as well. And for these, you can change the colors to represent what is high density and what is low density. These are not as common in publications, but is definitely an option to display your data by density. You can also display your data by expression in a certain channel. So the first way is via a dot plot by heat map. So in this case, you can look at 3 parameters in one graph. So here I'm looking at CD4 positive and CD8 positive cells. And this is the same dot plot, but I adjusted the heat map to look at expression of CD45RA and CD45RO in this population. So just in these 2 dot plots, I can look at 4 targets. And that allows you to see where you might find those CD45RO cells without looking directly at that parameter. Another way by expression is by histogram, and this allows you to view 1 parameter at a time, but it does give you relative cell number between negative and positive for that parameter. So the histogram on the left is showing you the x-axis. So all of these cells on the left will fall to the negative, and all of the cells on the right will fall to the positive. And we can see, without even looking at percentages, that there are more cells that are positive for CD4 than negative. And then similarly with CD8, all of the cells that are on bottom will fall on the left or the negative side because we're looking at the y-axis. And then all of the cells on top on the y-axis will fall into the positive. And we can see that there are less CD8 positive cells than there are CD8 negative. So these are 2 examples of showing your data by expression of a certain target or more than one target. You can also plot just simple events based on their expression of these markers. It won't show you the density and it won't show you expression levels of anything else, but it is a nice clean way to show where your cells fall between 1 or 2 parameters. And dot plots and histograms are quite common in publications as well. Speaking of histograms, there are many ways that you can show these, especially if you want to compare populations. So if you drag samples on top of one another in your layout editor, it will create an overlaid histogram. You can also do this with dot plots, which I'll get to in a little bit, but you'll have a few more options if you use the histogram format. And that would be found when you double click on the histogram, and you can change whether it's half offset, so you can see the samples a little clearer. Same thing with offset, and they're all in one graph here, which is nice. You can also stagger offset, although I will warn against this. It does tend to skew your perception of the data because in this case, it looks like the sample in blue has higher expression of the target when really they are about the same as the red when you look in the other histogram layouts. So just be careful with the stagger offset. And you can also shade 1 or more of your histograms to draw attention to 1 or more of your samples. So your sample of interest, and if it needs to be in black or white, you can change the line style or the line weight, again, to draw attention to 1 or more of your samples that is of interest. In terms of dot plots, you can change the information that's shown on these plots. So you could have the name of the population and the percentage, just the percentage or just the name or nothing, depending on if you want quantifiable data. And that can be changed when you double click on the graph, and you can choose to show the frequencies or the percentages in that gate or the population name of that gate. You can also choose to show ancestry. Ancestry just shows all of the gating steps that it took to get to the population you're showing. You can also choose to show adjunct histograms. So this is where that overlay dot plot comes in handy. If you overlay 2 samples over one another, the dot plots may not show as much if they are very similar, but this will create a histogram on each axis representing that data. And this looks about the same because there's only 2 populations here, but this may be a nice way to show your various populations or your various samples and compare them in more than one way. You can also compare them with visible words or statistics, and that can be directly in the legend, so attached to the graph itself. And so when you look at the legend, there's a few places where you can add this information. The first one is going to be in the add parameter column, and this is based on the gating that it took you to get to this point. So you can add the information on which compensation matrix was applied to each of these samples, or the annotation in the workspace. The full gating path can also be helpful information when you're showing data. So these are something that will then display another column on your legend, giving that information for each of the samples in the overlay. You can also add statistics here in your legend. One that is very common in publications and talking about each experiment is MFI, or mean fluorescence intensity, and that's how brightly your cells are stained for a certain parameter. So in this case, if I add MFI for my channel, which is correlate plus 488, and that falls into FL1 or FITSI channel, we can get the MFI of this channel, and we can compare statistical numbers between the populations instead of just using visual cues. And so this gives a little bit more data, and that you can plot in different types of graphs in other software. Finally, you could add the keyword that was applied to this. So that's another way that keywords come in handy. If I have an overlay of a lot of samples and I have a keyword that I wanted to show which sample belongs to which keyword, I can do that. So in this case, I added both the MFI of these samples and my keyword. So I added a keyword titled donor, and I assigned all of my samples to either donor 11 or donor 12, and this is able to tell directly which sample is from which subpopulation of that keyword. So now I want to switch gears only slightly to talk about how you could make your data look more presentable and more appealing just to the eye. And the first is through axis transformation. And you might notice that when you're analyzing, sometimes dot plots or histograms can look scrunched, or they could have extra space on one side, and you can adjust how these cells fit on the axes via this transformation tool. So you can transform either the x-axis or the y-axis on the plot that you're looking at, and it will change it for the entire workspace. So when you click on transform, you can customize the axis, and that will generate a histogram of the axis that you're looking at. And when we look at that, we can also change the scale. The most common is by exponential, but you can also change it to any of these other options if that is a better way to display your data for your needs. But I'm going to be showing the options that by X gives you, because your scale will determine which of these tools you can use. So for the most common scale, you can use the positive and minus arrows on the positive side of the axis to change how much of your graph is showing. This would have been very helpful for me in grad school. I didn't know this existed, and I repeated a lot of experiments because FlowJo was telling me that I had 30 to 40% of my events on the chart edges. If I were to just increase this axis, I would have been able to find those cells. It just means they're fluorescing more brightly than what I'm showing on my graph at that time. You can also change and move around these scroll bars, and they will change various aspects of your graph. So width basis will change how wide the bottom of your cells are. So if they're very wide, you could make it thinner so that your cells look more like a cohesive population. But every experiment is different, so you may want to play around with these and click save a few times to see how that changes your data until you get to a place that you like. Additionally, you can change your axis information directly in FlowJo in this case. So I would a lot of times export my images and then put them in PowerPoint and adjust the axis titles that way, but you can attach it directly to the image so you know what you're looking at every time. And you can type in whatever information you'd like to display on your graphs in the layout editor. You can change whether you can see the numbers on the axis, whether you can see the axis at all. And this is helpful for whatever needs you may have to show your data. Like I said earlier, if you want something quantifiable, or if you just want something visual to show you have expression of a certain marker, to draw attention to a specific part of your data. Also in FlowJo, you can line up your graphs without having to squint and stare at them for 5 minutes straight, which I did a lot. So I didn't know these tools existed, but they've been here. So I wanted to pass on the knowledge in case other people didn't know that these tools were here. So you can align your graphs either on the left or the right so that they're directly on top of one another. Same thing with aligning them on the top or the bottom or vertically, so that all of your graphs are in a straight line. And almost more importantly, you can space them evenly, either vertically or horizontally, so that there's an equal amount of space between each of your graphs, which makes it just look more appealing to the eye. And so these tools are very helpful. Like I said, it takes about 30 seconds to do this instead of 5 minutes for each 2 graphs to make sure they're lined up. So that's definitely something that has helped me save a lot of time. And so in summary of everything that I've showed you today, most importantly, it's good to set yourself up for success from the get-go. And that happens by knowing your machine very well and to take time while building your panel. That can only help you when you're analyzing so that you don't have to go adjusting the compensation matrix a lot. And you don't have to repeat experiments because you have too much overlap between colors. So taking time when you build your panel will help tremendously when it comes to analysis. Speaking of controls, they're also very important, almost as important as your stained samples, because again, this allows you to gate properly to avoid bias, and it allows you to use more parameters in your experiment so you can get as much data out of your one cell population as you possibly can. And this at the bottom is just an example of one of our products that we've released recently, which is a basics panel. So this is a panel that we validated to work well together. And I used all of the tools that I've showed you today to make this data look presentable and look appealing to the eye. And so I just wanted to represent that here because setting yourself up and using the right controls can get you a lot of information from the cells that you want to know so much about. And so with that, I'd be happy to take questions. And I think Afrida has some additional slides to share with you about Proteintech and some related products. So I'm going to stop sharing now.
Thanks so much, Michelle. That was a really wonderful talk, very comprehensive. As Michelle said, I do just want to highlight a few products that might be beneficial for our audience today. So the first products that I want to highlight are our conjugated primary antibodies. So Proteintech offers primary antibodies that are directly conjugated to our Correlite Plus dyes. And our Correlite Plus dyes cover a wide range of wavelengths for easy multiplex panel design. They also have unmatched brightness and photostability and are equivalent to Alexa Fluor in spectral profile. All our conjugated antibodies undergo stringent validation so that you can be confident that you will obtain reliable results. And because of their quality and performance, they are perfect for precious samples and low abundant targets. Proteintech also offers antibodies that are conjugated to conventional dyes for flow cytometry, including FITC, PE, and APC. We also offer Atlantic Blue, which is spectrally similar to Pacific Blue, and Cardinal Red, which is spectrally similar to Alexa Fluor 594 and Texas Red. And so sometimes we know that it's difficult to find a pre-conjugated antibody that we like or that fits our panel, but Proteintech offers a solution for that too with our flexible antibody labeling kits. Flexible is a novel antibody labeling kit that uses an affinity linker to conjugate fluorochromes, enzymes, and molecules in any buffer condition. The protocol for Flexible is very easy and it only takes 10 minutes. So within 10 minutes, you can label your primary antibody, and this is regardless of the supplier of the antibody. Because you're pre-conjugating your antibody beforehand, it is perfect for same species multiplexing. And also because it is so simple and fast to use, Flexible allows for you to easily rearrange your panel. I also want to highlight a new product that we just launched, which is our flow cytometry panels, which Michelle talked a little bit about at the end of her talk. Our flow panels are perfect for those who are just beginning flow or someone who is looking for the convenience of a pre-designed and pre-optimized panel to help them find their target cells. Our panels use top-sided clones that you know and trust and include 5 antibodies that are conjugated to different fluorophores with the PE channel open for your target of interest. Currently, we have these 6 kits here in the lower left-hand corner, which have been designed and optimized by our experts in-house. And finally, Proteintech also offers supporting reagents and kits for flow cytometry, including isotype controls, secondary antibodies, viability dyes, cell health assays, and fix and perm buffers. Before the Q&A starts, I also just want to mention one more thing, which is that as an attendee of today's webinar, we will be sending a follow-up email with a link where you can choose one free full-size flow antibody conjugated to our Correlate Plus dyes from the list that you see here on the right. We will send this list in the email as well. We will also provide a certificate of attendance as well as the recording for this webinar. Please allow a few days for this email to be sent to your inbox. So with that, I'll go ahead and I will begin the Q&A session. If after the Q&A session you still have any questions, please do feel free to email our events team at events@ptglab.com, or you can even email Michelle directly at michelle@ptglab.com. So I know that we have some questions. So the first question that I see is, how would you make sure of diagonality of cells? Do you just draw that yourself?
Yeah, for that, I just draw it myself. There should be a pretty clear diagonal, but it's always helpful when you go through all of your gating steps to go back to that gate if you feel like you have cells in there that don't look right, or maybe double positive when you don't expect to see that. That could be cells that are stuck together that you accidentally gated into the rest of your gating. So I would recommend using that as something that can be changed as you go through gating to reflect what you want. But yeah, it should be a pretty clear diagonal. And again, you might have to use those transform axes tool to make it look as clear as possible.
Great. We have another question asking, so in regards to compensation with beads versus cells, do beads account for cell size and complexity as well as expression level of our protein of interest, or are those parameters not relevant for the purpose of calculating compensation?
Yeah, that's a good question. I used to think it had to do with the cells as well, but it's all about the dye in the case of compensation. So you just want to tell the machine, this is what this dye looks like based on the voltages that you've set. And so the cell complexity or target expression levels don't matter. It's all about the dye. In that case, the dye is what stains the beads, not the antibody. So half the beads will bind to the dye and half won't, and that you can get a very clear answer on what a positive in that channel would look like.
Great, thanks. We have another question asking, can you elaborate more on what MFI is describing and what form of MFI is best to report? So median, geometric mean, algorithmic mean. Sorry about that.
Yeah, that is a tricky question because it depends so much on what you're trying to reflect. I would warn against comparing MFIs between experiments. So if you run flow on 2 different days and you run the exact same experiment, the exact same parameters, you might get different numbers. And so in that case, I wouldn't necessarily report it if you ran the experiments on different days. But if you're running a few samples and you want to compare those samples in terms of that parameter, it really depends on whether you expect to see a positive and a negative population or just the positive, or just negative, I guess. Geometric mean will help you when there's two populations account for the logarithmic axis or the exponential axis. So that will take into more account the positive cells that are a lot brighter than the cells sitting at 0. But that's a lot of statistics involved in that, and it depends on what you want to show with that data. But like I said, try and avoid using MFI as a strong statistic for inter-experimental reporting.
So the next question's asking about, so in your presentation, of course, you show a lot of examples of FlowJo, since that's what we use at Proteintech, but they were asking whether there are other softwares that have the same formatting options.
That I don't know about. I'm really only familiar with FlowJo. I did a little bit of digging to see what other softwares offer. And it seems like based on the company that you're using, they will have user guides to the different tools that they offer. So even FlowJo users, I recommend going through that, because there's a lot that I didn't know when I first started using FlowJo that would have helped me. And there's probably stuff that I still don't know about FlowJo. So I would recommend looking at the user guides for your analysis software to see where you might find those tools, and maybe even other tools that I didn't touch on that might be helpful to you.
Great, thanks. So there's a question about adding graphs from the layout editor. So they're asking, how do you add your graphs from the layout editor into a Word document? I have had some problem with blurry graphs in the past.
Yeah, my recommendation for that is, instead of taking a screenshot of your layout editor, because that's what I used to do, I would recommend exporting the image. So on FlowJo, it's under File, and you can export everything that's in that layout as whatever file format you'd like. So normally I use JPEGs because that has a high quality. So if I use a JPEG, then I can drag that directly into the Word document and format that however you want. That should get rid of blurriness, although I can't promise anything. That could just be based on your computer too. But usually JPEG will give you a clear image if you export the image directly instead of screenshotting it.
Can you use an FC block in your sample prior to antibody labeling instead of isotype controls?
Yeah, so you can use isotype controls to block as well. I didn't really touch on that. I was talking about isotype controls more as a separately stained sample to use in gating. So that was not in the same sample as the stained sample. That was just to account for any background from the dye. But yes, FC block is always recommended to make sure that your binding is specific and you're not getting non-specific binding to the FC receptor by your antibodies. So I would always recommend using some sort of blocking agent. It seems like you can use isotype controls, but FC block is also very helpful. But avoid using FC block if you are staining for any of the targets that are in those compounds. Usually it's CD16 and 32 for mouse, or 16, 64, 32 for human. So if you're looking for any of those targets, I would recommend using an isotype instead of FC block.
So the next question's asking, what's the difference between using unstained and FMO as a control?
Yeah, so unstained samples are great for telling you where your cells are in the first place, but an FMO is helpful because you can get to those really rare populations to make sure that your gates are drawn in a precise way. So in the example I showed, I was looking for a very rare population in the blood. So I had an FMO control that was stained for everything except that rare marker, so that I could get down to that rare population and then look for expression of that marker. Unstained control may not give you the same ability to be precise when drawing that gate. So if you have a rare marker or a very large panel that you're looking at one thing or two things in particular, an FMO is really helpful in those cases.
So this is kind of a product question for you. So do all of our products work with any flow machine?
That is a question that is probably better for your flow machine, actually. So when you're purchasing a flow cytometer, you'll have various options for the lasers that are in your machine and the filters or detectors that are a part of that machine as well. And so you can look at all of the different options. There are endless options for these configurations. And based on the dyes that you want to use, you would pick a machine that can do that dye. But I would also recommend going onto some sort of spectra viewer, or Fluorofinder has a great tool that you can find through our website a link to. And you can type in the machine you have, and then also what dyes you're using, and you can view the spectra that way to make sure that your machine can detect those dyes.
Great. So do you have any recommendations of a free software, or do you know whether there's any free software or plugin for FlowJo for spectral data, particularly making spectral ribbon plots?
I don't have a ton of experience in spectral flow, unfortunately. I do know that there is some software that can be used with FlowJo. As for a plugin, I'm not so sure about that. I do know that a lot of people who run large panels using spectral flow tend to use more of a coding omics based analysis software because FlowJo tends to crash a lot from what I hear. So it might be worth looking into a different analysis software if you're having issues with FlowJo, just so that your data is very clear. But like I said, I don't have a ton of experience with spectral flow, so I don't know what FlowJo offers specifically.
Our next question is asking, when and how do you use the trigger threshold option? So their device in particular is Cytoflex S, and because they work with exosomes, is it often recommended?
Um, I might have to look into that. I don't know a lot about trigger thresholds, and I have done no research on exosomes. So I can look into that and see what I can find. But as of right now, I don't know much about that topic.
Okay. And then it seems like there is one more question that I see. So do you mind recommending an anti-rat FC option or an anti-mouse or anti-human applicable? If you have
That
anything.
really depends on the cells that you're using. So if you are staining rat cells, then yeah, I would recommend using an anti-rat FC block. But if you're using human cells, I would recommend a human one, and same thing for mouse. There might be some products out there that can block any FC receptor, but usually the clones are different in those products, and they're specific for the clones that have been found in rats to block the FC receptor. So yeah, I would try and use the species of the cells that you're staining.
It seems like there is one more question that I missed. It's about our flow cytometry panels. Do you know if there's any plans for to do marine samples?
Yeah, we are currently working on some mouse panels. I don't know exactly when they'll be released, but we're working behind the scenes on creating some good basics panels for anti-mouse.
Great. All right, I think those were all the questions. If I missed your question, I really apologize. Please do feel free to email Michelle. She's very friendly, as you can see, and she is very responsive. So if you have any other questions about events, please do email us at events@ptglab.com. And if you liked today's event and would like to know more, please do follow us on our socials, because we do post about our events there. So with that, I would just like to thank Michelle once again for the very informative presentation, and thank you to all our attendees for attending this webinar, and hope everyone has a great day.